First, search for the protein sequence on the NCBI database. It might be easier to find it first on NCBI than on PubMLST. Select a full-length sequence specific to the species you are interested in. You can download the sequence as a text file in FASTA format or copy and paste from the page. When you have the sequence, go to PubMLST. In the Neisseria typing database, select Single Sequence. Paste in the amino acid sequence for the protein of interest. Press Submit. A hit will be returned for any similar sequences within the database. The locus tag is shown here. The protein coding sequence can also be displayed. Clicking on the locus identifier will then bring up its description. From the Neisseria species database, select the genome collection. From the drop-down menu, select Species. In the text box, type in the species you are interested in. Press Search. At the bottom of the page in the Analysis Tools section, select Export Dataset. In the Provenance section, include any information you want to include in the output, including country. From the drop-down list under Loci, find the locus you identified previously for your gene of interest. Press Submit. When the analysis has completed, you can export the search results as an Excel file. The Excel sheet shown the isolate ID, country of origin, as well as allele or gene variant. Sort the data according to allele, and then country. You will see, there are a large number of isolates with identical alleles from the same country. You can edit the list so each country has, say, around 4 representative isolates with the same allele. Later on, when you make your gene tree using the Genome Comparator tool, you will need to have edited your list to no more than 4,000 isolates. It's quite a big list, so this might take a little while. When you have edited your list, go to the Analysis tab in the Neisseria Isolate database. Scroll down the list of analysis plugins and select the Genome Comparator tool. Launch the Genome Comparator. Enter in the isolate identifiers from your edited Excel sheet. The total number should not exceed 4,000. Enter in the locus ID for the gene of interest. At this point you can add in additional information to include in the output, including country. Press Submit. When the job has completed, scroll to the bottom of the page. Click on the tab marked Distance Matrix, Nexus Format. The distance matrix will be displayed on a web page. Save the web page rather than copy and paste, this will keep it in the Nexus format. You will be able to open the Nexus file in Splits Trees. From Splits Trees you can make changes to the tree. You can also save the tree data in a format suitable for loading into the Interactive Tree of Life website. I will include a video on the Interactive Tree of Life on another day.